Hello team, let's look at the first question. So drawing deoxythymidelic, so I just put the sugar here and the phosphate group, that makes the acid, right? Now we need to put the base and the numbering, right? In this case, I need the thymine, so I'm just going to put the pyrimidine, the smaller single ring, right? So now if you just copy the structure, so this is how it should look like. Now the numbering, super important, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. That's for the sugar. And make sure it's the 2 prime deoxy, so get rid of the OH at the 2 prime. So no hydroxyl group there. Now labeling the thymine 1, 2, 3, 4, 5, 6 in this order, and no prime, but fifth position has a methyl. So that gives you thymine, right? But if you kill the methyl group, and that will give you the uracil, right? Okay, now this is important. So numbering and labeling, if it is triphosphate, that would be alpha, beta, gamma, all 3 phosphates there. So think about that, and then now whatever else you have to make, you can go from here, right? So that is the thymidelic acid, and it's deoxy, right? That's the simple D for that. Now we can just get rid of one piece at a time, right? So you lose the phosphate pieces, and then the phosphate linker, so that's actually the ester bond. Then you will have the nucleoside, the sugar and the base. Now you lose the sugar, you will have only the nitrogenous base, right? So, but make sure you draw them though, right? I'm just showing you like how the steps should be, right? Your process, it's easier. You start with the bigger, you end up with the smaller, right? Just chop, 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 go to the final, whatever the question asking. Now you lose the methyl in the 5th position, so you will get the uridine, right? So that's the way you want to answer this one. Okay, all right. Now we can go to the other example. Yes, if you make the uridine, make sure 2 prime is a hydroxyl group, right? Now we go to the sequencing steps. So in this case, the target DNA, to be sequenced, if needed, you're gonna do multiplication by PCR, right? So let's say we have a hair sample, blood sample, something like a nasopharyngeal swab or saliva. So we're probably gonna multiply the DNA once extract from the cell, and so we have plenty of DNA. Now we need to have primers. So normally the kit comes with the primers, or we can just design the primers. That's only to be the 3 prime end, and there should be basically 2 types of primers, right? So you added the primers, and then we go to the next step, right? Now the prime annealing is a little bit lower temperature, okay? So because those are short pieces of DNA, right? So it's hard to bind otherwise. Now so we move on to the next one. So we're gonna add the DNA polymerase, and we have to add DNTPs. So it's something like Taq polymerase, right? And then we have to add the DNTPs. So all DNTPs, these are basically, actually, if you can visualize it, it's 4 different channels or 4 different columns, right? And then they will be added, and with the traces of the terminator, right? So this is full concentration, and it works perfectly with the polymerase a little bit higher temperature, and now the polymerization takes place. Now we have to add the traces of the terminator. So the terminator is the DDNTP. So in all 4 channels we have 4 different DDNTPs, right? So that means DDATP, DDGTP, GGCTP, and so on. Now make sure if you are able to recognize that, you are able to put the numbers and sequential letters if needed. Okay. Now the next step is actually now we have so bunch of DNA fragments. Some of them are very short, some of them are longer, some of them with the blue tag, the red tag, the yellow tag, and so on, because these are coming with the fluorescent dye, right? So because they are attached to the ddNTPs, right? So we're going to see the color coding. So now I have basically 4 different bunches of different sizes of DNA, right? So these ones we can separate and analyze using the gel electrophoresis. So that's the next step. And so different sizes, different colors based on the dye, right? And fluorescent dye. And then now we just put them into the gel electrophoresis, right? And we should get the, let's remove these ones. Okay, here. So my gel, let's simply show like there are 4 wells and then 4 different bunches coming in, and then they will run, and then the smallest ones goes, each portion goes into each well, and then the smallest ones go very, very bottom, because the positive is positive pole is in the bottom, right? And then the tiny ones go to the bottom and the bigger ones stay on the top almost, right? And then we start, basically, I mean, you can logically think you start from the smallest DNA piece in the very bottom, and then you just work your way up, or you just put it to the data analysis, right? So the data analysis will feed into a computer process, and the program will tell you what's the sequence, right? But in the good old days, this is how we do it, right? So we just go from the smallest and work your way up and analyze, and then we know that. Okay, so the next question is, so this is a standard one. Look at this one. So you have to know if it is not explained, you typically you type 5' to 3', and it's a coding strand given, then I should be able to draw my template, right? So you simply follow the Watson-Crick pairing. So A to the T, T to the A to the T, G to the C, G to the C, and so on. You have to continue until you complete this one, right? And then from the other end too, so you can go like that. So now I have the double helical DNA, right? Now typically what you do, so that's the coding, this is the template, so recognize them, and now we can actually denature the double helix into 2 single strands, but I'm just going to put it as it is, and then so you can see the primers has to be only 3' direction to 5' direction, right? So we're going to just write the primers for it, right? So I select the 3' end, right? And let's put it here. This 3' end needs a primer that should look like 5'. So T to the A to the T, T to the A, C to the G, and so on. So that's 1'. So similarly, then you can continue as long as you want, right? So in this case, we have to write up to 15, right? And then you're going to do the same thing up here, 3' end of the other side of the coding, and then that you start with the G to the C, A to the T, C to the G, and so on. So then now you have 2 primers, right? And that's the goal. Okay, so most of you actually answered this question very nicely. And if you didn't, just follow the video, stop the video and try yourself first, and then practice a bit. So key steps in cloning with using the bacteriophage and lambda, right? In this case, we're going to use actually the quality of infectious quality of the bacteriophage. So this goes and attacks on the bacteria, so we can use the lambda page to deliver the DNA we need. So what we need, actually, we take the lambda DNA and we actually keep its portion of the blueprint, the coding that needed for the infection, but we can remove some of the other parts, right? So we cut with the restriction enzymes, and keep a middle area open. So it's probably about 50 base pairs, right? Now we have, let's say, human DNA or plant DNA, something special that we need to incorporate. So that's the target DNA, right? So that one also ready with the sticky ends. We cut with the restriction enzymes, the particular ones, and then now we are good to put together the two Lego puzzles, right? So you can combine them and ligate them, and then we can create the chimeric DNA, right? So now we have the page DNA carrying our target DNA, and we just have to deliver that to a cell, right? So this is the target DNA, and then it's now in between it's incorporated into the lambda page DNA. Now we're going to deliver this one back to the lambda page, and then that will infect the bacteria, right? So that can transform. So package into the page, and that will go to that will infect the bacterial cell, and then we can grow that into a culture and select the colonies we want, right? And then we can grow large scale, right? And then you can go further analysis and PCR and other things too, right? Okay, so that's the sort of the guide for doing some of the questions. And if you have particular problems, just email me. So I will do another one for the today's some of the discussion, because some of you folks didn't show up. So I'll put that one too here. Okay, thank you.